Good day everyone. Welcome to your biology review for college entrance test in D-shape. Biology questions test our understanding of living systems and not just memorization. In exams, one correct answer is often hidden among the structures that sound right but aren't. So in this review where it will help you understand why the correct answer is correct, see why the other choices are wrong so you don't get tricked, and gain test-taking confidence by practicing elimination skills. Now, I'll be giving you 5 tips to answer biology questions in college entrance tests. First one is, of course, read carefully. Watch for words like always, except, and best describes. Next one, eliminate wrong choices. Narrowing down improves the odds of getting the correct answer. Next, use examples. Of course, we can link concepts with real-life cases so that we can better understand what is the question talking about. Next is associate words or associate it to keywords and manage your time. Do not get stuck in one question. Return to hard questions later. Okay? So let's start. Let us have question number 1. Organisms develop features that help them adapt better to their surroundings. What property allows them to survive in their habitats? Okay, we have 4 different choices here: order, organization, regulation, or homeostasis, growth and development, and evolutionary adaptation. You can pause the video for you to try to answer it first before we reveal the answer. Okay? The correct answer for this one is evolutionary adaptation. Why is it the correct answer? Populations change over generations, so traits that improve survival become common. So that's why that's the correct answer. What are the keywords from the question? Some of the keywords from the question would be organisms develop features that adapt to their surroundings and survive. So when we're adapting and surviving, that talks about evolutionary adaptation. Why not A? Okay? Order means cells and structures are organized, but it doesn't guarantee survival. Why not letter B? Homeostasis maintains stable conditions inside. Okay, inside one organism, and it's not a long-term adaptation. It's only for a short period of time. Why not letter C? Growth and development explains how the individuals will mature, but not about how the species will adapt to the environment. Okay, so you can see some pictures here in the presentation. You can pause the video for you to check on these concepts for you to further understand what is it talking about. Okay, now let's go to question number 2. Hybrids in the wild are rare and usually arise only between closely related species. More often, a species passes on its traits, producing organisms similar to itself. What property of life does this illustrate? Now, from the stem or from the question, let us get some keywords from the question. So, our keywords will be passing on traits and producing similar organisms to itself. Okay, so the choices will be growth and development, evolutionary adaptation, reproduction, and metabolism. Now, what is the answer here? Our answer, the correct answer will be reproduction. Okay, why? What is reproduction? Why is it the correct answer? Reproduction is passing traits to offsprings, ensures continuity of a species. Okay, so reproduction is the one that ensures that the species will continue its lineage across generations. Why not A? Okay, growth and development is about one individual increasing in size and not producing offspring. Okay, why not letter B? Evolutionary adaptation happens over many generations and it's not a single reproductive event. Okay, why not letter D? When we say metabolism is about the energy use. Okay, and there are 2 types, the catabolism and anabolism. And it's not about passing on the traits from one individual or one generation to the next generation. Okay, next one, question number 3. Which of the following is true regarding the role of phospholipids in biological membranes? Okay, so first choice, we have phospholipids are hydrophobic and form the inner layer of the membrane. B. Phospholipids are primarily involved in the storage of energy in membranes. C. Phospholipids are hydrophilic and orient towards the inside of the membrane. D. Phospholipids form a bilayer with hydrophilic heads facing the inside and outside of the cell. So what will be the answer here? Okay, so the answer will be letter D. Okay, phospholipids form a bilayer with hydrophilic heads facing the inside and outside of the cell. Okay, so why? Why is it the answer? The heads of the membrane face water on both sides while tails hide inside. Okay, because we have what we call hydrophobic and hydrophilic parts of the cell membrane. Why not letter A? The inner part isn't a whole layer, but it's only the hydrophobic tails. And then why not letter B? Energy storage is a role of triglycerides and not the phospholipids. In this case, we are talking about phospholipids. Now, why not C? Okay, only the heads are hydrophilic, the tails inside are hydrophobic. That's why they are hiding inside. They don't want to interact with water. Okay? Now, question number 4. What protein structure level is a beta-pleated sheet? A. Primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. The correct answer here is letter B, secondary structure. Okay? Why is it secondary structure? So when we're talking about the structure of proteins, we can classify them into 4. Okay? The primary protein structure, secondary protein structure, tertiary protein structure, and quaternary protein structure. Okay, primary structure is only an amino acid sequence, as you can see in the picture. Okay? It's a chain, one long chain of amino acids. Okay? Now, for secondary protein structure, this is the time that they form alpha helices and beta sheets. Okay? That's what we call the alpha helix and the beta pleated sheets. If you can see from the picture, beta pleated sheets are the ones that are folded. It looks like it's folded. But for alpha helix, it's the one that looks like it's coiled, like the structure of a DNA. It looks like a structure of the DNA. Okay? So, for tertiary protein structure, it's the overall 3D folding of one chain. Okay, again, it's just one chain, but there is a 3D folding of it. It may include beta-pleated sheets and alpha helices. Okay, for quaternary structure, it's the interaction of multiple chains. Okay, so it's now the protein consisting of more than one amino acid chain. So it can also contain beta-pleated sheet and alpha helices. So in this case, the answer is letter B, secondary structure. Now let's go to the next question. Which of the following is a characteristic feature of saturated fatty acids? A. They are found in higher concentrations in plant oils. B. They are typically in a form of liquid at room temperature. C. They have no double bonds between carbon atoms in their hydrocarbon chains. D. They have one or more double bonds between carbon atoms in their hydrocarbon chains. Now, what will be the answer here? Okay, so the correct answer will be letter C. They have no double bonds between carbon atoms in their hydrocarbon chains. Why? One characteristic of unsaturated fatty acids will be the double bond. Okay, so if you will see a double bond in the hydrocarbon chains, that will indicate that it is an unsaturated fatty acid. So that's what we call the point of unsaturation. Okay, why not letter A? Okay, plant oils is mostly unsaturated. So that means when it's unsaturated, there is a presence of double bonds. Okay? Why not letter B? Liquids at room temperature are unsaturated oils. Okay? So they're typically in a form of liquid at room temperature. That means they are unsaturated oils or unsaturated fatty acids. Why not letter D? Double bonds will always indicate unsaturation. That's why we call it point of unsaturation. So the correct answer is letter C. Okay, saturated fats are full of hydrogens and usually solid. So they don't contain double bonds. Okay, now let's go to question number 6. How is the arrangement of two strands of DNA running in opposite directions described? Okay, so first choice is double helix, B anti-parallel orientation, C covalent bonding, D complementary base pairing. Okay, the answer here is letter B, anti-parallel orientation. Let's get a keyword from the question. Just in case we don't really know the answer, let's get some keywords from the questions. What do you think are the keywords from the question? Some keywords that we can have is opposite direction, running in opposite direction. So from that case, we can look for context clues that have the same meaning in the choices. First one, double helix. Why not double helix? Okay, double helix describes the spiral shape and not a direction. Okay, it describes the structure of the DNA. Why not letter C? Covalent bonds links nucleotides in one strand and not a strand orientation. It's like a bond between molecules and it does not pertain to a strand orientation or a direction of a strand. Why not letter D? Base pairing or complementary base pairing explains that adenine is always paired with thymine, as you can see in the picture, and cytosine will always pair with guanine in a DNA structure. Okay? So, it's a matching, it's not directionality. So, from the choices given, we can say that the only choice that is related to the question is anti-parallel orientation. In an anti-parallel orientation, one strand of the DNA runs from 5' to 3' and the other strand of the DNA runs from 3' to 5' orientation. So that's what we call the anti-parallel orientation. Okay? Now let's go to the next one. Which part of eukaryotic cell performs protein synthesis? Okay, from the word itself, protein synthesis, that means it is the making of proteins. Okay, so first choice, rough ER or rough endoplasmic reticulum. B, smooth endoplasmic reticulum. C, Golgi apparatus. And D, mitochondria. Okay, so what really is the answer? Okay, so which part of eukaryotic cell performs protein synthesis? Okay, it's really the ribosomes. Okay, ribosomes is the one that helps in protein synthesis. But miss, there are no ribosomes from the given choices. Okay, so when you are taking entrance test, we cannot tell the proctor that, oh, there's no correct answer in this one. So they're not gonna make it bonus for everyone. So what's the best thing that we need to do? The best thing that we need to do is choose the best answer that we can get. Okay? So the answer here will be rough endoplasmic reticulum. Why? What's the reason why that's the best answer? It is because ribosomes are attached to the surface of rough endoplasmic reticulum to synthesize the proteins. Okay? Not necessarily all the time because there are ribosomes that we can see in the cytoplasm, but most of the ribosomes are attached in the rough endoplasmic reticulum. So that will be the best choice that we can choose. Okay? Why not letter B? Smooth ER makes lipids and detoxifies. Okay? There are no ribosomes in its surface. Why not letter C? Golgi apparatus modifies and packages proteins and doesn't make them. Okay, but they help in packaging the proteins, but not in the synthesis of proteins. Why not D? Mitochondria, of course, we know that one is the powerhouse of the cell, but let's revise our understanding. It's the one that generates ATP. What is ATP? It's adenosine triphosphate. Okay, it is a source of energy and not a protein. Okay? So, I hope that is clear for everyone. Now, let's go to number 8. Which type of fiber makes up the cytoskeleton composed of tubulins? Okay, so from the question, we can infer that the answer is microtubules. Okay, because of the word tubulins. And that is a correct guess. That's a good guess. Okay, but let's take a look. A, microfilament, B, microtubule, C, intermediate filaments, D, extracellular matrix. Now, the correct answer will be microtubules, of course. We can have context clues. We can get tubulins, so we can infer that it's microtubules. Now, let's take a look at the reason why microtubules is the answer. Microtubules are built from tubulin dimers, okay, and it's important for transport and division in the cell. Okay? Next one, why not letter A? Microfilaments are not tubulin-based, but they are actin-based. Okay? So, microfilaments are actin-based. It's formed with filamentous actin. Next one, why not letter C? Intermediate filaments give structural support and it is not made of a tubulin. And why not ECM or the extracellular matrix? Extracellular matrix, from the word itself, extracellular, that means it is outside the cell. Okay? So when we say cytoskeleton, it can be found inside the cell. Okay? So ECM is not a cytoskeleton element. Okay? Let's go to question number 9. Which of the following is a special type of passive transport exhibited by water molecules on selectively permeable layers? Okay, so we have here our choices: phagocytosis, diffusion, osmosis, and facilitated diffusion. What do you think is the answer? Okay, the answer is letter B, osmosis. Why is it the answer? When we say osmosis, it's a passive water movement through membranes, as you can see in the picture. Okay? So, osmosis is a movement of solvent from low to high solute concentration in a semi-permeable membrane. Okay? Again, osmosis, movement of solvent. Okay? What is our solvent here? Water. Okay, coming from low concentration to high solute concentration to achieve equilibrium in a semi-permeable membrane. Okay, why not letter A? What does that mean when we say phagocytosis? Let's take a look at the picture. Phagocytosis is active cell eating. Okay, that's what we call it, cell eating. So that means it engulfs solid particles going inside the cell. Okay, to create phagosomes. Why not letter C? When we say diffusion, it's the movement of solutes and not specifically the water. Okay, it's more of a movement of the solutes and not the solvent. Why not letter D? Facilitated diffusion. Facilitated diffusion is a type of diffusion wherein proteins are used, okay, to help move the solutes and not the water. Okay, so when we're talking about water molecules moving from low concentration to higher concentration of solutes in a semi-permeable membrane, it is osmosis. Okay? Next one, number 10. What are glucose and oxygen considered in cell respiration? A, reactants, B, products, C, catalysts, D, enzymes. Okay? Now let's take a look at the formula on the right. So this is the general formula for cell respiration. In cell respiration, our body is creating ATP or energy. That is the goal, to release the ATP and energy. Now, this one happens in the mitochondria. Remember, mitochondria is what we call the powerhouse of the cell or the site for ATP synthesis. Okay? So, for cellular respiration to happen, we need glucose. Mitochondria needs the glucose coming from the food that we are eating, oxygen coming from the air that we breathe, and this will react. These two will react. So, what are the formula of glucose? C6H12O6. Formula for oxygen gas is O2. We need these two in order for it to react, okay, so that it can create ATP, which is the energy released, carbon dioxide, okay, that's the one that we exhale, right? And then water that is used by the body. So this is the general formula for cell respiration. Now, the question is, what are glucose and oxygen considered in cell respiration? In a chemical reaction, there are two sides. We have the reactants and the products. The reactants are the ones that need to create a change or do chemical change in order for it to create the products. Okay? The new set of chemicals. So, oxygen and glucose in cell respiration on the left side of the equation, we call it the reactants. Okay? Now, why not letter B? Letter B is products. Okay? Products of cell respiration are ATP, carbon dioxide, and water. Okay? What about catalysts? When we say catalysts, these are enzymes. Of course, examples of it are enzymes, and they speed up reactions but are not consumed. Okay? Unlike in reactants. Now, enzymes control steps, but glucose and oxygen aren't enzymes. Okay, so they are reactants. Okay, next one, let us go to number 11. Which of the following is an electron carrier? Okay, now let's take a look. Electron carrier, what are we talking about? This is the cellular respiration. Okay, so the choices are pyruvic acid, acetyl coenzyme A, NADH, and ATP. Let's have a quick background. In cell respiration, there are three main steps. First one, we have the glycolysis, then the Krebs cycle, and then we have the electron transport chain or the oxidative phosphorylation. Okay, take note of that. Oxidative phosphorylation. Now, the answer here is letter C. Okay, letter C is NADH. Why is it the electron carrier? When we say pyruvic acid is a product of glycolysis, as you can see in the picture. Okay, in glycolysis, glucose is transformed into a pyruvic acid and it is not a carrier. Okay, it is a product of glycolysis. Now, why not acetyl coenzyme A? Acetyl coenzyme A delivers the acetyl groups into the Krebs cycle. Okay? That's the one that delivers the acetyl groups going inside the mitochondria for it to be able to undergo Krebs cycle. They are not electrons. Okay? Acetyl coenzyme A are not electrons. Next one, why not letter D, ATP? ATP stores energy but it does not carry electrons. Okay? It stores the energy. Now, why is it NADH? There are actually two electron carriers in cell respiration. Okay? After the Krebs cycle will produce 2 ATP. Okay? Aside from the 2 ATP that the Krebs cycle will produce, it will also produce NADH and FADH2. Now, these two are the electron carriers that will bring it to electron transport chain or the oxidative phosphorylation. So, that's why we call it electron carriers because it's needed by the electron transport chain or oxidative phosphorylation to create more ATPs. Okay? So, since in our choices, there's only NADH, the correct answer is NA.